Right now, somewhere beneath the surface of soil you've never thought about, something is happening that has been happening for hundreds of millions of years. A root is deciding what to let in, not with a brain, not with anything that resembles a decision in any way you'd recognize, and yet the outcome is selective, and the selectivity is real, and the consequences of getting it wrong are serious enough that entire species have been shaped around getting it right. Welcome to the Sleepy Biologist. If you're here for the first time, this is a place for long, unhurried explorations of biology, the kind that ask nothing of you but to rest and listen. If you've been here before, it's genuinely good to have you back. A warm thanks to channel members and everyone who supports through Super Thanks. This channel exists because of that kindness, and it's felt. If tonight's topic moves something in you, or if you're listening from somewhere interesting, the comments are always open and always read. But for now, just settle. You don't need to do anything. The biology will come to you. There's a zone around every living root, not visible, not dramatic, called the Rhizosphere. It's not a layer you could point to or lift away. It's more like an influence. The chemistry a root releases into the soil immediately around it changes what's available, what dissolves, what moves closer. Microbes gather there in concentrations far higher than in the surrounding soil. Ions shift. Organic acids seep out from the root itself, quietly altering the pH of the nearby environment. The root, in other words, is not passive before the first cell wall is even reached. It is already reshaping the conditions of what it will encounter. You do something similar before a difficult conversation, rearranging the space, adjusting the air. The root does this continuously, and it does it chemically, and it has no idea it's doing it. What surrounds a root in contaminated soil is not simply dirt with something extra added. Heavy metals, cadmium, lead, arsenic, zinc in excess, nickel, exist in different forms depending on soil chemistry. Some are tightly bound to soil particles, some float loosely in soil water, some have formed compounds with the organic matter that make them temporarily inert. The rhizosphere, with its altered pH and microbial activity, can shift a metal from one of these states to another. A metal that was bound can become free. A metal that was harmless in its current form can become something the root now has to manage. The root's own chemistry sometimes increases the availability of the very things it will then have to decide about. This is where selectivity becomes important. The root's outer layer, the epidermis, is not a passive membrane. Water moves into it primarily through specific protein channels. Mineral ions move through transporters, proteins embedded in the cell membrane that are shaped for particular ions. Potassium has its transporters. Calcium has its pathways. Iron, when the soil is iron poor, triggers the root to release compounds that pull iron toward it and then take it up through dedicated proteins. The system is specific because specificity was necessary. Taking in everything indiscriminately would have been lethal. Plants evolved in soils full of things that could damage their cells, and the transporter system is one answer to that problem. Heavy metals complicate this answer. Cadmium, for instance, is not something a plant needs. There is no biological function that requires it. But cadmium is chemically similar enough to zinc, an element roots genuinely need, that it enters through zinc transporters. The protein cannot distinguish between them. From the transporter's perspective, the ion has the right shape, the right charge behavior, and so it is carried inward. This happens in most plants. It happens in plants you've eaten. The selectivity of the root is real, but it's not perfect, and it was never designed to manage an industrial world. Arsenic enters through a different case of mistaken identity. In soil water, arsenic often exists as arsenate, a form that is structurally similar to phosphate, which plants need for nearly every energy-related process inside the cell. Phosphate transporters in the root membrane accept arsenate. They don't choose to. They simply respond to a molecule that fits. In most plants, arsenic entering this way quickly interferes with cellular chemistry. The damage is real and measurable. But in certain species, and this matters for everything that follows over the next few hours, the interference doesn't stop the plant. Something else is happening alongside the uptake, something that changes what the arsenic encounters once it's inside. The root tip itself, just millimeters long, is where most of this selectivity is concentrated. Older root tissue becomes covered in a layer called suberin, a waxy, water-resistant compound that reduces the movement of ions through the cell walls of the root's outer layers. In young, unsuberized roots, ions can move through the spaces between cells as well as through the cells themselves. As the root ages, that pathway closes. The apoplastic root, the path through cell walls and intercellular space, is restricted. More and more what enters the root has to pass through a membrane, which means passing through a transporter, which means the selectivity of those proteins becomes the primary filter. There is a structure called the Casparian strip, a band of suberin that rings each cell in a layer called the endodermis, deep inside the root, just outside the vascular tissue. Everything heading toward the plant's interior has to cross the endodermis, and the Casparian strip forces all of it through the living cell membrane. Nothing gets through the wall anymore at this point, only through the cell. This is the plant's last checkpoint before the vascular system, and it is the place where what has entered the root either continues inward or gets stopped. In plants that accumulate metals, what happens at this checkpoint is different from what happens in plants that don't, and that difference is part of what makes phytoremediation possible at all. The soil keeps offering, it doesn't stop. Ions continue arriving at the root surface, moving by diffusion and by the plant's own chemical influence. The root continues its quiet work, absorbing, excluding, managing, sometimes failing to exclude what it should have blocked. Somewhere between what the soil contains and what the plant eventually holds in its leaves, a negotiation plays out that neither party controls completely. The metals that make it through are not there by force. They entered through channels the plant built for other reasons, in a world that didn't include them. You might recognize something in that. Being built for one set of conditions and finding yourself in another. Processing things your original design didn't quite account for, managing anyway. The root doesn't know what it's handling, it only knows how to handle it, or how to try. And in certain plants, the trying has become, over an enormous stretch of time, something closer to mastery. What happens after the metal crosses the endodermis and enters the vascular system is a different kind of story. It moves faster there, carried upward in the water. The plant is pulling through its whole body, and what waits for it in the cells above is already preparing. There's something worth noticing before anything else. The cell wall of a plant root is not alive in the way the cell inside it is alive. It's a structure, built by the cell surrounding the cell, made mostly of cellulose fibers woven into a mesh. And yet what passes through it and what doesn't shapes everything that follows. It's not entirely clear how to think about this. A wall that isn't making choices, but whose properties determine outcomes. A structure with no intention that nonetheless behaves in certain conditions like a filter. You notice it when you look at what the wall actually contains. Cellulose fibers make up the main body of the wall, but running through the spaces between them are other compounds, pectin, hemicellulose, various proteins. Pectin in particular carries a negative charge under normal soil pH conditions. Negative charges attract positive ions. Most heavy metal ions in soil solution, cadmium, lead, copper, zinc in excess, carry positive charges. They are drawn electrostatically toward the cell wall before they've even reached a membrane. This isn't absorption in the way a cell absorbs something. It's more like accumulation at a surface. The metal sits there, held by charge, not inside the cell, not outside the root entirely, somewhere in between, in a zone that is neither fully controlled nor fully uncontrolled. In plants that don't accumulate metals, this can be a stopping point. The lead arrives at the wall, binds to pectin, and stays there. It doesn't cross the plasma membrane, it doesn't reach the cytoplasm. The cell wall functions, in this case, as something close to a buffer, not designed for this, but useful for it. What's less certain is how much capacity this buffer has. The binding sites on pectin are finite. In soils with high metal concentrations, or in roots that have been exposed to contamination for a long time, those sites fill. Once they're saturated, the metals that keep arriving have nowhere to pause. They reach the plasma membrane directly. And the plasma membrane is a different kind of boundary entirely. It's a lipid bilayer, two sheets of fat molecules arranged so their water-repelling tails face inward and their water-attracting heads face out. Ions cannot cross this on their own. Water molecules move through dedicated channels called aquaporins. Mineral ions move through transport proteins, each shaped for specific ions or specific chemical conditions. The membrane is selective in a way the cell wall isn't, not by charge, but by structure, by the specific shapes of the proteins embedded in it. This is where the mistaken identity problem becomes most visible. A transport protein shaped to carry a particular ion does not inspect the ion. It responds to the ion's size, charge, and chemical behavior. If another ion shares enough of those properties, it may be carried too. There's something almost uncomfortable about this when you sit with it, a system that evolved across enormous time, refined by countless generations of selective pressure, and it still gets fooled. Not always, not catastrophically in most conditions, but consistently enough that in contaminated soil, the cellular interior of most plant roots contains metals that were never supposed to be there. It's not entirely clear whether supposed to is even the right way to think about it. There is no blueprint. There is no original intention. The transporter proteins exist because ancestors that had them survived better than ancestors that didn't, in the soils those ancestors grew in. The soils most plants evolved in didn't contain cadmium at industrial concentrations. The transporters weren't wrong, the conditions changed. What happens at the plasma membrane in a hyperaccumulator, a plant that actively concentrates metals rather than simply failing to exclude them, is still being studied in detail. Some research suggests these plants have modified transporter proteins with slightly different binding properties. Others point to the sheer number of transporters expressed in their root cells, a higher density of uptake capacity. Some work suggests the interior chemistry of the cell in these species is different enough that metals enter more readily, because the concentration gradient pulling them inward is steeper. The cell is already processing and moving them away from the membrane faster than other plants do. Each of these mechanisms is real. They may all be operating at once. The cell wall holds what it can. The membrane carries what fits through its channels. The cell receives what arrives at its interior and begins immediately the work of managing it. In a hyperaccumulator, this management is different from what happens in a plant that can't tolerate metal load, and the difference starts, it seems, almost immediately after entry. There's still quite a lot that isn't fully resolved. Which transporters dominate in which species? How much of the uptake is passive, driven by concentration gradients, and how much involves active transport that costs the plant energy? Whether the cell wall's buffering capacity can be modified by the plant itself in response to contamination, or whether it simply fills until it can hold no more. What is clear is that the boundary isn't one thing, It's several things, each with different properties, operating in sequence. The metal in contaminated soil doesn't push its way in. It moves through a series of zones, each with its own chemistry, its own capacity, its own way of either slowing or permitting passage. By the time something has crossed all of them and reached the interior of the root cell, it has already travelled through more selectivity than most biology ever asks a molecule to pass through. The cell that receives it is already doing something in response. What exactly it does and how long it can keep doing it without cost is a quieter stray, one that moves upward through the plant's body slowly, the way water moves. Not in a rush, following the same pull that everything inside a plant follows, toward the light the leaves are waiting to happen. There is a particular quality to the way arsenic moves through a plant cell that stands apart from how most harmful things behave in biology, and it has to do with the fact that arsenic, once inside, does not announce itself. It substitutes itself quietly for something the cell was already using, and the damage that follows is not the damage of invasion, but the damage of replacement, of a process continuing almost normally but not quite, of an outcome that looks right from a distance and is wrong in every detail up close. Arsenic in its arsenate form enters the root cell through phosphate transporters, as described before, carried inward because its chemical shape is close enough to phosphate that the transporter protein does not distinguish between them, but the resemblance to phosphate does not end at the membrane. Inside the cell, arsenic continues to behave like phosphate in ways that matter enormously, because phosphate is not simply a nutrient the cell stores, it is a working part of the cell's energy system, a molecule that participates actively in nearly every energy transfer the cell performs. The currency of cellular energy is adenosine triphosphate, ATP, a molecule whose energy is stored in the bonds between its three phosphate groups. When the cell does work, building proteins, moving ions across membranes, repairing damage, it breaks one of those bonds and releases the energy held there. What remains is adenosine diphosphate, which the cell then recharges by attaching another phosphate group, restoring the molecule to its 3-phosphate form so it can be used again. This cycle runs continuously in every living cell, at a rate that's difficult to hold in mind, thousands of times per second in active tissue, driven by the chemical reactions of cellular respiration. Arsenic can enter this cycle. It can bind where phosphate binds, participate where phosphate participates, and form compounds analogous to ATP. The problem is that the bond arsenate forms in these compounds is unstable in a way that the phosphate bond is not. It breaks spontaneously, without releasing energy in a usable form, without producing the recharged ADP that the cycle requires. The cell performs the motion of energy production and captures nothing from it. The machinery runs and the output is absent. This is what stands out most about arsenate toxicity. Not that it stops the cell violently, but that it keeps the cell moving while quietly draining the return on that movement. In most plants, this process is damaging enough that arsenic accumulation beyond a certain point becomes lethal. The energy deficit compounds. Processes that depend on ATP slow, then fail. The cell begins to lose the ability to maintain the concentration gradients across its membranes that keep everything in the right place. Ions leak where they shouldn't. Water follows. The cell swells or shrinks or simply becomes unable to perform the ordinary work of being alive. What happens in arsenic hyperaccumulating species, and the best studied among them is a fern, Pteris vittata, which can accumulate arsenic in its fronds at concentrations that would destroy most plant tissue, is that this sequence is interrupted much earlier. The arsenate that enters the cell is reduced enzymatically to arsenite, a different chemical form of arsenic, and this reduction happens quickly enough and thoroughly enough that the arsenate does not spend long in the phosphate mimicking form that disrupts ATP synthesis. Specific enzymes, arsenic reductases, perform this conversion, and in Pteris vittata these enzymes appear to be expressed at high levels in root tissue, meeting the arsenic soon after it crosses the membrane. Arsenite, the reduced form, is less reactive than arsenate. It doesn't mimic phosphate. It doesn't insert itself into the energy cycle in the same way. What it does instead is interact with sulfur-containing molecules, particularly the thiol groups found in certain proteins, and in a small molecule called glutathione. And this interaction is the beginning of the containment process, rather than the beginning of the damage process. The cell has effectively changed the nature of what it's dealing with by changing the arsenic's chemistry before the arsenic has time to cause the disruption it would otherwise cause. This is what stands out most when looking at how hyperaccumulators handle arsenic. Not just that they tolerate it, but that they change it. The arsenic that exits the root cell and moves into the vascular tissue of Pteris vittata is predominantly arsenite, already transformed from the form that entered. The plant has not simply accepted a toxic load. It has processed the toxin in transit and sent a chemically different version onward, one it is better equipped to manage in the tissues that receive it. The extended picture of arsenic moving through this plant, entering as arsenate, converted to arsenite in the root, carried upward through the xylem in that converted form, and eventually sequestered in the cells of the frond, is a picture of a process that works because each step feeds into the next, because the chemistry at one stage creates conditions that make the next stage possible, and because the whole sequence runs without visible sign of the effort involved. The fronds of Pteris vittata in a contaminated field look like fronds. They are green and structured and continue to photosynthesize. The arsenic inside them at concentrations that in other species would have collapsed every cellular membrane sits in vacuoles, contained, chemically bound to other molecules, removed from the active chemistry of the cell. The extended metaphor of arsenic as an imposter, slipping through by resemblance and then being caught and changed before it can fully assume the role it was mimicking, is one way to hold this process in mind, and it's not entirely wrong, but it eventually becomes less useful than simply noticing what the chemistry actually does, which is more interesting than any metaphor for it. The arsenic reductase finds arsenite and changes it. Glutathione and related compounds bind to arsenite and carry it toward the vacuole. The vacuole receives it and holds it. The cell around the vacuole continues. What the cell continues doing, photosynthesizing, growing, dividing in the frond's developing tissue, while holding this internal cargo is the part that remains after the chemistry is laid out, genuinely worth pausing on. The arsenic found something when it entered. It found a cell that had been, across a very long stretch of evolutionary time, learning what to do with it. The finding in the end was mutual. The leaf doesn't know what it's carrying. That's the first thing worth sitting with. Not because it's strange. Biology rarely knows anything in the way we mean when we use that word, but because the leaf looks so ordinary, green, flat, arranged to catch light, doing exactly what leaves have done for hundreds of millions of years, which is to hold themselves open and let photons move through their internal chemistry and become slowly sugar. And inside that leaf in certain plants there is cadmium, or arsenic, or nickel in concentrations that would dissolve the cellular machinery of most living tissue, sitting in vacuoles, held in chemical bonds, not moving, not causing visible harm, just there in the cells, in the spaces the cell set aside for it, while the rest of the cell continues its ordinary green work. You start to notice when you think about it slowly that storage is not a passive thing. It had to be built. The vacuole, that large internal compartment, sometimes occupying most of the cell's volume, is not simply an empty space where unwanted things collect. Its membrane, the tonoplast, is studded with transport proteins specifically tasked with moving particular ions from the cytoplasm into the vacuole's interior. The interior itself is maintained at a different pH from the cytoplasm, more acidic, which alters the chemistry of what enters and helps keep certain metal complexes from drifting back out. The vacuole is not passive storage. It is active containment maintained continuously, requiring the cell to keep spending energy on the problem of keeping the arsenic away from the machinery the arsenic would otherwise interfere with. Past, first. The leaf had ancestors, millions of years before this particular fern or this particular alpine pennycress grew in soil that humans contaminated. Related species were growing in naturally mineralized ground. Outcrops of serpentine rock rich in nickel and magnesium, volcanic soils with unusual chemistry, ancient seabeds lifted into mountains and slowly weathering into substrate. The plants that survived there, that reproduced there, that eventually came to dominate those difficult patches while other plants died or refused to establish, they were accumulating. Not because accumulation was a plan, because the cellular machinery that happened to move metals efficiently into vacuoles happened to protect the cytoplasm from damage, and protection meant survival, and survival meant offspring, and offspring meant the machinery was passed forward. The leaf of a hyperaccumulator today carries in its cells a history that long predates the industrial contamination we now use it to address. Something in that feels worth noticing quietly. Present now, the leaf is expanding. In the meristematic tissue at the frond's tip, the actively dividing zone, new cells are forming, and as they form they inherit the tonoplast transporters from their parent cells, the same proteins, the same capacity for sequestration. The metals moving upward from the root in the xylem stream arrive at these developing cells and are taken in and moved quickly to the vacuole. The young tissue accumulates faster in many hyperaccumulators than the older tissue does. The newest cells are, in some species, the most active in uptake. Growth and accumulation are happening simultaneously in the same tissue, without one visibly slowing the other. You start to notice that the leaf is not being poisoned and surviving. It is growing into a system already prepared to receive what the soil is sending up. The metaphor that almost fits here, of a house built around a difficult thing, architecture shaped by what had to be contained, gets close, but then the biology moves past it, because the leaf didn't plan its architecture, and the metals didn't choose their location. And what actually happened is more interesting than any human construction metaphor can hold for long. The chemistry is quieter than the metaphor. In the vacuole, metals are often found bound to organic acids, malate, citrate, oxalate. These acids are produced by the cell's own metabolism. Ordinary byproducts of the chemistry cells perform regardless of metal load, and in hyperaccumulators they serve an additional role as ligands, molecules that bind to metal ions and stabilize them in a form less likely to react with other cellular components. The metal arrives. The organic acid meets it. A complex forms. The complex sits in the acidic interior of the vacuole, relatively inert, relatively stable, while outside the vacuole's membrane the cell's cytoplasm maintains its normal chemistry. Relative. It's always relative. The containment is not perfect. There are conditions, cellular stress, drought, damage, under which the vacuole's membrane becomes less stable, under which some of what was sequestered can leak back into the cytoplasm. The maintenance is ongoing because the problem is ongoing, and in very high accumulation conditions there are measurable signs of cellular cost even in hyperaccumulators. Subtle changes in enzyme activity, shifts in which genes are expressed, slight reductions in growth rate. The leaf holds its stillness, but the stillness is effort, and then the metaphors dissolve, and what remains is just the cell, the vacuole membrane doing its work, the organic acids holding their metal cargo, the chloroplasts in the same cell catching light and fixing carbon, and doing the photosynthesis. The plant needs to keep funding the energy cost of containment. All of it in the same small space. The leaf doesn't know what it's carrying, but carrying it, the leaf shaped itself around it, and the shaping goes back further than the contamination does, back to soils that were difficult before difficulty was something we measured. That history is in the leaf right now, in the vacuole, in the tonoplast transporter moving one more ion inward quietly, while the light does its separate and ancient work just a few organelles away. Soil is not uniform with depth. This seems obvious when stated, but the implication of it takes a moment to settle. A root growing downward through 30 centimeters of ground is not moving through a consistent medium. It is moving through time. The surface centimeters of soil are young. They turn over through freeze and thaw, through earthworm activity, through the decomposition of fallen organic matter that brings last season's chemistry into contact with this season's roots. What sits in the upper layer of a contaminated field may reflect something deposited recently: atmospheric fallout from a nearby smelter, surface application of sewage sludge used as fertilizer decades ago, irrigation water carrying dissolved metals from upstream. Recent contamination tends to concentrate near the surface because the processes that would move it downward are slow. Below that, something older. Lead moves through soil slowly, very slowly. In most soil types, lead binds tightly to organic matter, to iron and manganese oxides, to clay particles. Its mobility in soil water is low compared to cadmium or zinc, which travel more freely with water movement. What this means is that lead contamination from, say, the decades when leaded gasoline was in widespread use, ending in most countries in the 1980s and 1990s, is still largely sitting where it settled. In urban soils, in roadside soils, in the soils of old orchards where lead arsenate was used as a pesticide through much of the 20th century, the lead that arrived then is still there now, held in the top 30 centimeters of soil in concentrations that have barely moved. Cadmium behaves differently. It is more soluble, more mobile. Rainfall leaches it downward. In acidic soils, it moves faster still. Contamination introduced at the surface can, over decades, migrate through the soil profile in ways that lead cannot. A root reaching 40 centimeters down in a cadmium-contaminated field may encounter metal that was at the surface 20 years ago, that moved incrementally downward through successive wet seasons, that now sits in a layer the root has only recently reached. Still, arsenic has its own movement pattern, different again. In oxidized soils, arsenate, the form arsenic takes when oxygen is present, binds readily to iron oxides, the rust-coloured minerals common in many soils. It stays put. In waterlogged or reduced soils, where oxygen is depleted, arsenic becomes more mobile, shifting into arsenite and releasing from iron oxides into soil water. Seasonal flooding, irrigation patterns, and the respiration of soil microbes can change the redox chemistry of a soil layer and mobilize arsenic that has been bound for years. A single wet season can bring arsenic into the soil water that wasn't there the previous year, at depths where roots are actively growing, presenting a metal load the plant hadn't been managing before. The root grows into all of this without a map. It follows water gradients, oxygen gradients, the chemical signals of nutrient-rich zones. It branches where conditions allow branching, and slows where the soil is too compacted or too toxic for the root tip to advance. The expiration is not random, but it is also not planned. It's a response system, following present chemistry, moving toward what the present chemistry suggests is worth moving toward. and what it finds depends enormously on the specific history of the ground it's entering. There's something in that worth sitting with for a moment. The idea that the root and the contamination are meeting across different timescales. The contamination arrived at one moment in history, from one source, and has been doing whatever chemistry dictates since then for decades, binding and releasing and migrating and transforming. The root arrives now following a different logic entirely, and encounters not a single problem but a layered record of a problem, each layer slightly different in chemistry, concentration, and form. What the plant eventually accumulates in its tissue is a kind of summary of that record. In studies of hyperaccumulators growing in contaminated fields, researchers have found that the metal profile in plant tissue often reflects soil heterogeneity, that plants growing a few meters apart in the same field can show meaningfully different accumulation levels, because their roots happen to explore different patches of soil history. One plant's roots reach the deeper cadmium-rich layer, another's remained mostly in the upper zone. The field looks uniform from above. The biology beneath it is not. Zinc in most soils is naturally present at some concentration. It's an essential micronutrient for plants and for most soil organisms. The contamination layer in a zinc-polluted site is often difficult to separate cleanly from background zinc levels, because the background exists, because the metal was never entirely absent. What changes with contamination is concentration and form, and both of those vary with depth and with the history of the site. What was deposited, when, in what chemistry, and what the soil has done with it since. The root descends into this variation. It extends into zones where the contamination is high and into zones where it's lower, and it takes up metals at different rates depending on what it finds. The phytoremediation happening in a real field is therefore not a uniform process drawing metal evenly from all depths. It is a patchy, depth-dependent, root distribution-dependent process, shaped by the same heterogeneity that characterizes all soil biology. Models of phytoremediation efficiency have to account for this. How deep are the roots reaching? What is the metal distribution with depth? What form is the metal in at each layer? And how does the rhizosphere chemistry of this particular species alter that form? The calculation is not simple, and the outcome in any given field is genuinely difficult to predict without detailed soil profiling. But the roots keep descending, following water, following gradients, entering layers of soil time that they were not grown to interpret and do not need to interpret, encountering what is there and doing what they do with it. The contamination waits in the dark, distributed through decades of soil history, and the roots move through it the way roots have always moved through difficult ground, without urgency, without a destination. Reaching because reaching is what roots do. Most plants die in contaminated soil. Not dramatically, not all at once. They germinate sometimes. They send roots into the upper layer, and something in the chemistry slows them. Growth becomes uneven. Leaves yellow at the margins. The root system stays shallow, stunted, unable to extend into the deeper soil where water might otherwise be found. Eventually the plant stops producing new tissue. It doesn't collapse, so much as cease. This is the normal outcome. It tends to be overlooked because the plants that survive in contaminated sites are visible, and the ones that fail to establish are not. What survives is worth looking at carefully. Tolerance in plant biology has a specific meaning. It refers to the ability of a plant to maintain relatively soluble function in the presence of a potentially damaging substance. It doesn't mean the substance isn't there. It doesn't mean the substance isn't doing anything. It means the plant is doing enough in response to keep going. I tend to think this distinction matters more than it first appears to. An immune system that neutralizes a pathogen before it causes any effect is doing something categorically different from an organism that experiences the pathogen's effects and manages to stay functional despite them. Hyperaccumulators are not immune to heavy metals. They are tolerant of them in the specific sense. They experience the metal load, they respond to it continuously, and the response is what keeps the damage below the threshold of lethality. The response has several components. One is exclusion at the root, limiting how much enters in the first place. Some hyperaccumulators show modified transporter expression that reduces uptake of the most damaging forms of a metal. This is partial. It reduces load without eliminating it. One is chemical conversion, as with arsenate being reduced to arsenite before it can interfere with phosphate chemistry. This happens quickly in root cells before the metal has time to act on the energy system. One is chelation, binding the metal to molecules that neutralize its reactivity. Glutathione, phytochelatins, organic acids. These compounds surround the metal ion and change what it can do, not removing it, changing it. And one is sequestration, moving the chelated metal into the vacuole where it stays. All of these run simultaneously. They run continuously, they cost energy. ATP, the same currency the arsenate disruption was targeting in the previous chapter. The plant is spending resources to manage what it accumulated, while also photosynthesizing, growing, reproducing. The management is not free. In studies comparing metal-tolerant plants to non-tolerant relatives growing in clean soil, the tolerant plants sometimes show lower growth rates under normal conditions. Not always, not dramatically. But the machinery of tolerance has a baseline cost that appears even when the metal isn't there, because the genes encoding that machinery are expressed constitutively, constantly, rather than only in response to metal presence. The plant is always ready. Being always ready has a price. What I find most interesting, and it's worth saying this gently, is that tolerance didn't evolve as a single adaptation. It's a set of overlapping mechanisms, each with independent evolutionary histories, that happen to work together in species that became hyperaccumulators. The arsenate reductases, the phytogelatin syntheses, the vacuole transporters. Each of these exists in some form in many plant species. What differs in hyperaccumulators is not the presence of entirely novel biology, but the degree of expression, the coordination, the efficiency of the system as a whole. This is reassuring in a way. The tools for tolerance were already present, distributed across the plant kingdom, doing other jobs. In certain lineages, under certain pressures, those tools were amplified and coordinated into something that looks, from the outside, like a specialized and unusual capability. And it is unusual. But it grew from ordinary biology, the same biology present in less extraordinary plants, turned up in particular directions by particular pressures. You can think of it less as the evolution of something new, and more as the refinement of something already there. The tolerant plant in contaminated soil is not performing a miracle. It is doing more at higher intensity of things that living cells have always done to one degree or another. It is maintaining its chemistry under conditions that would overwhelm the same mechanisms in a less refined system. It keeps going not because it is unaffected. It keeps going because the response is fast enough, thorough enough, and sustained enough that the effect never fully outpaces the management. There is something in that, in the idea of a continuous, costly, imperfect management that nonetheless keeps a living thing functional, that feels quieter and more honest than the word tolerance usually suggests. The plant is not fine, it is managing, and managing in the right soil with the right machinery turns out to be enough. The next question isn't whether the plant can survive the metal, it's what specifically happens to the metal once the plant decides, in the only way biology decides anything, to deal with it. What the chelation process looks like from the inside, what molecules are doing the binding, and why some of those molecules are the same ones your own cells use when they need to protect themselves from things they weren't built to handle. There is a molecule your body makes right now that plants also make, that nearly every living cell on Earth produces in some form, and it has been doing the same essential job since before animals and plants diverged from their common ancestor, which means it's been doing this job for somewhere close to 1 billion years. It's called glutathione. It's small as molecules go, 3 amino acids linked together, and one of those amino acids, cysteine, carries a sulfur atom with a particular chemistry that makes it, in the right conditions, extremely good at holding on to metal ions. You already know this feeling in a way, the feeling of gripping something tightly not to use it, but to prevent it from moving. Glutathione does something like this with heavy metals, not to use them, not to digest them or expel them immediately, simply to hold them in a form that can't react freely with the rest of the cell's chemistry. The process starts with sensing. Plant cells in metal-exposed tissue detect elevated metal concentrations through mechanisms still being studied in detail. It seems to involve the metals themselves interacting with existing proteins in ways that alter those proteins' activity, triggering downstream signaling. The cell doesn't have a dedicated metal detector in the way it has dedicated photoreceptors for light or dedicated receptors for certain hormones. The sensing is more distributed, more diffuse, multiple different cellular responses activating in parallel when metal load crosses certain thresholds. What follows the sensing is synthesis. An enzyme called phytogelatin synthase becomes active in the presence of metal ions, particularly cadmium, arsenic in its arsenite form, lead, and others. This enzyme uses glutathione as its raw material and links multiple glutathione units into longer chains. These longer chains are phytogelatins. Their structure gives them multiple cysteine residues arranged along the chain, and multiple cysteine residues means multiple sulfur atoms available for metal binding. Multiple grip points, each one holding the part of a metal ion complex. A single phytogelatin chain can coordinate one or more metal ions, holding them at specific positions along its length in a configuration that neutralizes their chemical reactivity. This is the part that's quietly worth sitting with for a moment. The phytogelatin doesn't destroy the metal. It can't. The metal is an element. Cadmium is cadmium, arsenic is arsenic, and no biological process changes one element into another. What the phytogelatin does is change the metal's context. A cadmium ion in the cytoplasm, free and reactive, will bind to sulfur-containing groups on whatever proteins it encounters first, including enzymes whose function depends on keeping those sulfur groups free. The cadmium disrupts the enzyme. The enzyme's function fails. Downstream processes that depended on that function begin to fail too. The cascade of dysfunction that heavy metal toxicity produces in sensitive plants is largely this: metals binding non-specifically to proteins throughout the cytoplasm, disabling them one by one. A cadmium ion bound to a phytogelatin is still cadmium, but its available binding capacity is saturated by the phytogelatin itself. It cannot reach the enzymes. It cannot bind the proteins it would otherwise disrupt. The chelation doesn't remove the problem, it contains it, holds it in the cytoplasm in a form that doesn't spread its damage to everything nearby, and then it moves. The phytogelatin metal complex is recognized by transport proteins on the vacuolar membrane, the tonoplast, and carried across into the vacuole's interior. Inside the vacuole, at the lower pH that the vacuole maintains, further chemistry occurs. Sulfide ions produced within the vacuole can join the complex, forming structures called phytogelatin metal sulfide clusters, larger, more stable assemblages that are even less likely to escape back into the cytoplasm. The sequestration is reinforced by the chemistry of the compartment receiving it. What was chelated in the cytoplasm becomes more stably bound in the vacuole. You've probably felt the impulse to put something difficult in a container and then add one more layer of containment, not from panic, just from the sense that one layer might not be permanent. The vacuolar chemistry does this automatically, without impulse, because the lower pH and the available sulfide happen to stabilize the complex further. It's the result of chemistry, not caution, but the result is the same. Phytochelatins are found in almost all plants and in many fungi and algae. Their presence isn't unique to hyperaccumulators. What differs in hyperaccumulators is partly the speed and scale of phytogelatin synthesis in response to metal exposure, and partly the efficiency of the downstream transport into vacuoles. A non-accumulating plant exposed to high cadmium will produce phytogelatins, but may not produce them fast enough, or may not transport the resulting complexes into vacuoles efficiently enough to prevent cytoplasmic damage before it accumulates. The machinery exists but runs below the threshold of adequate protection. In hyperaccumulators, the same machinery appears to be calibrated differently. Higher basal expression of phytogelatin synthase, more abundant vacuolar transporters, sometimes a larger vacuolar volume per cell providing more containment space. The difference between a plant that dies in contaminated soil and one that accumulates metals in its leaves without apparent harm may, in part, come down to the speed and capacity of a process involving a molecule only 3 amino acids long. The glutathione at the start of this is worth returning to. It was already there, doing other jobs, acting as a general antioxidant, protecting cell components from oxidative damage, participating in the detoxification of various compounds the plant encounters in normal soil chemistry. The phytogelatin synthase repurposed it, took the existing molecule and linked copies of it into longer chains specifically suited for metal binding. The cell didn't build a new tool, it extended an old one. Somewhere in the dark of the vacuole, a phytogelatin chain holds its cadmium quietly. The cell around it continues. The leaf grows. The contamination that entered at the root is now in a form that can travel with the plant, contained inside it, no longer in the soil, no longer available to the water table or the organisms that move through that soil. The chelation happened without light, without observation. It happens now in fields you will never visit, in plants that have been doing this work continuously for longer than the contamination has existed. Begin with something small, a single plant cell somewhere in the upper leaf tissue of a hyperaccumulator, a cell that's been alive for several weeks, that has been photosynthesizing through the daylight hours and resting through the nights, that has divided once since it differentiated from meristematic tissue and will likely not divide again. It is unremarkable in most respects. Its chloroplasts are arranged near the cell wall, angled toward incoming light. Its nucleus sits to one side, managing the ordinary traffic of gene expression that keeps the cell performing its functions. And occupying perhaps 60 or 70% of the cell's interior volume, pressing the cytoplasm into a thin layer between itself and the cell wall, is the vacuole. In plant cells generally, the vacuole is a dominant structure. It holds water, maintaining the turgor pressure that keeps plant tissue firm. It stores nutrients. It contains pigments in flower cells. It isolates compounds that would be damaging if left in the cytoplasm. Not just metals, but also defensive chemicals, secondary metabolites, compounds the plant produces to deter herbivores or pathogens. The vacuole in an ordinary plant cell is already doing considerable work. It is not simply empty space. In the cell of a hyperaccumulator, the vacuole is doing all of that and more. The metals that arrive via phytogelatin complexes, transported across the tonoplast by dedicated proteins, are now inside. Not dissolved freely in the vacuolar fluid, but held in complexes, bound to phytokeratins, to organic acids, sometimes incorporated into sulfide clusters. The vacuolar pH maintained around 5 or 5.5 compared to the cytoplasm's near neutral 7 alters the chemistry of these complexes in ways that reduce the likelihood of them releasing their metal cargo back across the membrane. The lower pH is not incidental. It is maintained actively by proton pumps embedded in the tonoplast, running continuously, pumping hydrogen ions into the vacuole to keep the interior acidic. This maintenance costs energy. It runs as long as the cell lives. There is a past to this arrangement. Vacuoles have been a feature of plant cells for as long as land plants have existed, and of eukaryotic cells for far longer. The endomembrane system, the network of internal membranes from which vacuoles derive, evolved over 1 billion years ago, initially as a system for internalizing material from outside the cell for digesting what was taken in, for sorting molecules to different cellular destinations. The modern plant vacuole is a descendant of that early compartment, repurposed over evolutionary time for roles its ancestors didn't perform. Storage, turgor-regulation, sequestration of secondary compounds, and in the lineages that became hyperaccumulators, sequestration of metals in quantities that would otherwise be incompatible with cellular life. The vacuole didn't evolve for phytoremediation. Nothing in biology evolves for a future use. The vacuole acquired the characteristics that make metal sequestration possible, because those characteristics were useful in the environments those plants actually lived in. Naturally mineralized soils, serpentine outcrops, volcanic substrates. That the same characteristics now allow certain plants to clean contaminated industrial sites is a coincidence of chemistry and circumstance, not a design solution. It is an odd coincidence, worth noting without overstating it. Back to the present, to the cell. The tonoplast transporters that move the phytogelatin metal complexes into the vacuole belong to a family of proteins called ABC transporters, ATP-binding cassette transporters. They are ancient proteins found in organisms across all domains of life, originally evolved for transporting a range of molecules across membranes. In plants, certain members of this family have been recruited for vacuolar metal sequestration. In Arabidopsis halleri, a small flowering plant that hyperaccumulates zinc and cadmium, specific ABC transporters on the tonoplast show dramatically elevated expression compared to closely related non-accumulating species. The protein exists in both, the expression level differs. What the vacuole holds can be imaged now with sufficient technology. X-ray fluorescence microscopy can map the distribution of metals within a leaf cross-section at cellular resolution, showing whether cadmium or zinc or nickel sits within the tissue. In hyperaccumulators, the signal concentrates inside cells, inside the vacuolar space, rather than distributed evenly through the tissue. You can see, if you have the right instrument, the containment that the cell has built. The metal is there, where the cell put it. The image, when described, sounds almost like evidence of intention. It is not. It is the outcome of chemistry selecting for chemistry, of cells that happen to sequester metals surviving in environments where cells that didn't were damaged, of that differential survival compounding over generations into a physiology that now reliably, across an entire species, does this thing that looks purposeful and is not. How long can it last? This is the uncertain part. The cell lives for weeks, perhaps months, before it senesces, before the cell begins the process of dismantling its own components in a controlled way, recovering what is useful and preparing the materials for redistribution within the plant. During senescence, vacuolar integrity can change. The tonoplast becomes less stable. The careful compartmentalization maintained across the cell's life may become less reliable. What happens to the sequestered metals during leaf senescence is an active area of research, whether they are remobilized into the phloem and redistributed to younger tissue, whether they stay in the senescing leaf and fall with it to the soil when the leaf drops, whether the outcome differs by metal and by species. In some studies, metals have been found to remobilize from older leaves to younger ones as the plant grows. In others, the metal stays in the oldest tissue and returns to soil with leaf litter. The answer appears to depend on which metal, which plant, and which growth conditions. It is not resolved in a way that allows a simple statement. The cell that has held cadmium quietly for several weeks may or may not release it when it dies. The vacuole that maintained its chemistry, that kept the proton pumps running, that held the phytochelatin complexes inside for the life of the cell, at the end of that life, the fate of what it was holding is not fully written. This is the uncertain part. It matters for whether phytoremediation actually removes contamination or only moves it temporarily. It matters for how plants are managed in remediation projects. When they are harvested, what happens to the biomass? And it circles back to where this chapter began, a single cell living its ordinary cellular life around an extraordinary interior. The chloroplasts at the wall, the nucleus to one side, the vacuole occupying most of the space, patient in a way that is not patience at all, but simply chemistry, continuing because the conditions that maintain it continue until they don't, and then something else begins. There is a language plants use when something is harming them, and it is not a language of sound or movement, but of chemistry, molecules released into the air, signals passed cell to cell through tissue, hormones travelling through the phloem to distant parts of the plant that haven't yet encountered the problem. When a caterpillar begins eating a leaf, the damaged cells release compounds that travel to undamaged leaves and trigger the production of defensive chemicals there before the caterpillar arrives. When roots encounter drought, abscisic acid moves upward through the plant and reaches the stomata, the small openings in leaves through which gas exchange occurs, and the stomata close to conserve water. The plant doesn't experience these events the way an animal experiences harm, but it responds to them, chemically, with what looks from the outside like urgency. In a hyperaccumulator carrying high metal concentrations in its leaves, much of this response language stays quiet. That's the observation that takes a moment to settle. Not that nothing is happening. The chelation, the vacuolar sequestration, the transporter expression, all of that is happening continuously at cellular scale. But the plant-level signaling cascades that in non-tolerant species announce metal toxicity, the production of reactive oxygen species that damage membranes and trigger stress responses, the release of ethylene associated with cellular damage, the visible symptoms of chlorosis and necrosis in leaf tissue. These are largely absent in hyperaccumulators at metal concentrations that would activate all of them in a sensitive plant. You start to feel, when you think about it slowly, that what's missing is almost as informative as what's present. Reactive oxygen species, molecules like hydrogen peroxide and superoxide, produced as byproducts of normal cellular metabolism and also in elevated amounts during cellular stress, are part of the plant's signaling network as well as being potentially damaging. At low concentrations, they serve as signals, activating stress response genes, directing the cell's attention toward a problem. At high concentrations, they damage lipids, proteins, and DNA. The balance between signaling and damage depends on the cell's antioxidant capacity, its ability to neutralize reactive oxygen species before they accumulate to damaging levels. Glutathione, the same molecule that serves as the precursor for phytochelatins, is also a major antioxidant. This is not a coincidence exactly, or rather it is a coincidence of chemistry, that the molecule useful for chelation is also useful for neutralizing reactive oxygen species, but it means the two systems are connected. In hyperaccumulators, glutathione pools are typically elevated. The cell maintains more of it than non-accumulating relatives do. This elevated glutathione serves both functions simultaneously, feeding phytochelatin synthesis when metals arrive, and buffering reactive oxygen species before they reach signaling concentrations. The result is a cell that under metal load does not produce the reactive oxygen spike that would normally activate the broader stress response. The signal that something is wrong doesn't reach the threshold required to send the message. From outside the cell, from the perspective of the plant as a whole organism, the metal load in the vacuole is not registered as an emergency. It's not entirely clear whether this is the cause or the consequence of successful tolerance, whether the plant doesn't signal distress because it genuinely isn't distressed at the cellular level, or whether the suppression of the distress signal is itself part of the tolerance mechanism, preventing systemic responses that might redirect resources away from the sequestration work happening in the affected cells. It might be both, operating in a loop. The sequestration prevents damage. The absence of damage prevents the stress signal. The absence of the stress signal prevents systemic responses that would cost resources. The conserved resources support continued sequestration. The loop runs quietly, still. The stomata of hyperaccumulators growing in metal-contaminated soil generally remain open at rates comparable to plants growing in clean soil. This matters more than it might first appear. Stomatal opening is one of the most sensitive indicators of plant stress. In drought, in heat, in toxic conditions, stomata close because the plant is prioritizing water conservation and tissue protection over gas exchange and photosynthesis. In heavy metal stress in sensitive plants, stomatal conductance drops. The plant is, in a measurable and visible sense, shutting down. In Arabidopsis halleri growing in zinc-contaminated soil, stomatal conductance has been measured at levels comparable to the same plant in uncontaminated conditions. The photosynthetic rate shows similar resilience. The plant is fixing carbon at normal rates while sitting in metal concentrations that would close the stomata of a non-tolerant plant within hours. The absence of systemic stress response is visible in the gas exchange data. Numbers that will look like a plant in ordinary soil from a plant that is not in ordinary soil. What ethylene does in this system is interesting. Ethylene is a gaseous plant hormone involved in senescence, in fruit ripening, in responses to mechanical damage and pathogen attack, and in responses to heavy metal stress in sensitive species. Cadmium exposure in non-tolerant plants reliably increases ethylene production. Ethylene production is associated with leaf senescence and abscission, the dropping of leaves, which can be read as the plant shedding damaged tissue. In hyperaccumulators, cadmium-induced ethylene production is substantially lower. The plant is not initiating the shedding response that a sensitive plant would use to remove its most contaminated tissue. This is, from one angle, simply a feature of tolerance. The contaminated tissue isn't damaged enough to trigger the response. From another angle, it raises a question, because ethylene-driven senescence is also a mechanism of metal remobilization, potentially moving metals from old leaves toward younger tissue or toward the roots. If hyperaccumulators suppress this response, what happens to the metals when the leaves eventually senesce through normal developmental pathways rather than stress-induced ones? The question doesn't resolve cleanly here. What can be said is that a hyperaccumulator in contaminated soil presents, at the level of whole plant physiology, something close to a picture of normalcy. Growing, photosynthesizing, exchanging gases, not signalling distress in the ways that distress is signaled. The metal is inside in the vacuoles, held in complexes, managed by machinery that runs without announcing itself. The plant looks fine. The question of whether it is fine, whether the absence of distress signals reflects genuine cellular contentment or simply a system that has learned to keep its internal work invisible, is not a question biology has fully answered. Something is always still running underneath the quiet. Notice for a moment what it means for a plant to be native to a particular kind of soil, not adapted to it, native to it, born into it in the sense that its parent grew there and its parent's parent through enough generations that the word ancestor stops feeling adequate. The serpentine soils of California, the nickel-rich ultramafics of Zimbabwe, the copper outcrops of Katanga in the Democratic Republic of Congo, these are not places plants moved into and then learned to tolerate. They are places where, across immense stretches of time, the plants that could survive out-reproduced the plants that couldn't, and the ones that couldn't are gone from those places now invisible in the record, present only as the absence of their descendants. What remained is what you find there today. Serpentine soils form from the weathering of ultramafic rocks, rocks from deep in the Earth's mantle, brought to the surface by tectonic movement and then slowly broken down by water and air over millions of years. They are chemically unusual in ways that matter enormously to plant life. They contain high concentrations of magnesium and heavy metals, nickel, chromium, cobalt, and very low concentrations of the nutrients most plants depend on: calcium, phosphorus, nitrogen. The ratio of calcium to magnesium is inverted from what most plant physiology is calibrated for. The combination is hostile to most life, and the vegetation of serpentine outcrops reflects this, sparse, often visually striking in its austerity, dominated by species found nowhere else. The plants that grow on serpentine soils are not simply tolerant. They are, many of them, endemic, found only there on that chemistry, unable to compete effectively in normal soils where other species can outgrow them. The adaptation that allows survival on serpentine has, in some lineages, come at the cost of competitive ability in ordinary conditions. The plant is calibrated for the difficult environment and only for it. This is an old pattern in biology. Specialization enables survival in a niche and constrains it elsewhere, somewhere in those long centuries of selection, and the time involved is genuinely difficult to hold in mind, not decades but millions of years, generations stacking into geological time. The cellular machinery of metal tolerance was refined into what we now observe and measure: the tonoplast transporters, the phytohelatin synthase expression, the elevated glutathione pools, the modified transporter affinities in the root membrane. None of this appeared suddenly. None of it was the result of a single mutation that transformed a sensitive plant into a hyperaccumulator overnight. What the genetic record suggests, and this is still an active area of research, with whole genome comparisons between accumulating and non-accumulating species revealing new details, is that hyperaccumulation in different plant lineages arose independently, multiple times. The trait is not inherited from a single ancestral hyperaccumulator. It evolved separately in ferns, in flowering plants, in different families and genera, each time through a different combination of modifications to existing biology. The outcome, high metal concentrations in shoot tissue, looks similar across these lineages, but the specific mechanisms differ in their details. Convergent evolution toward a shared solution to a shared problem, using whatever molecular tools each lineage already had available. What stands out most in this picture is not the convergence itself, but what it implies about the underlying biology. If multiple lineages, starting from different places, arrived independently at something resembling hyperaccumulation, it suggests the cellular machinery required was not exotic or difficult to produce. The basic components, the transporters, the chelators, the vacuolar compartmentalization, were already there, distributed across the plant kingdom in their ordinary roles. Hyperaccumulation required not the invention of new biology, but the amplification and coordination of existing biology, happening repeatedly wherever the selective pressure was strong enough and lasted long enough. The contaminated soils of the industrial era are, in geological time, very recent. A few centuries of mining and smelting, 100 years of leaded gasoline, decades of industrial agriculture. These timescales are nothing against the millions of years during which serpentine endemics developed their tolerance. The hyperaccumulators we use for phytoremediation today did not evolve to clean up what humans made. They evolved for older problems, in older soils, under older pressures. We found them already equipped, already doing what they do, and redirected that capability toward a newer version of the same basic problem: soil with too much of something in it. There's something in this that wants to be overstated and shouldn't be. The plants are not our partners in remediation in any meaningful sense. They have no awareness of the project. They grow in contaminated soil because they can, not because of any alignment of purpose. The evolutionary history that produced their tolerance is not a history that included us. We arrived late to a process that was already old. And yet, the cellular memory of those millions of years is present in the root tip, reaching into cadmium-contaminated soil right now, in a field somewhere, doing what its ancestors did in natural mineral soils, not knowing the difference and not needing to. The arsenate reductase in the root cell of a Pteris vittata fern doesn't know whether the arsenic it's converting arrived from a naturally arsenic-rich geological substrate or from the runoff of a pesticide operation. It converts what arrives. It has been doing this, in one ancestral form or another, for long enough that the time involved has no human equivalent. The pressure nobody witnessed produced the tool we now use. What the contamination finds when it enters the root is not a plant that learned in any lifetime to manage it. It finds a plant that carries in its cells the accumulated response of geological time to the kind of problem contamination presents. The response is old, the contamination is new. The meeting in a field somewhere is happening right now. The serpentine outcrops that shaped those first tolerant lineages are still there, in California and Zimbabwe and elsewhere, still hosting their endemic species, still selecting, still running the slow process that built what we now borrow. The cellular memory holds. The plant has done its work. Roots extended into contaminated soil. Metals crossed the root membrane, were chelated, moved upward through the xylem, sequestered in vacuoles throughout the shoot tissue. The leaves accumulated what the soil released, and the contamination that was in the ground is now, in part, inside the plant. This is where most explanations of phytoremediation stop. It is not where the process stops. What happens next, after the plant reaches the end of its growing season, or reaches the biomass level the remediation project considers sufficient, determines whether any of the work was permanent. The plant is not the end point. The plant is a vehicle. What matters is what leaves the site when the plant does. If the plant is left in place, it dies. Decomposition begins. The microbes that break down plant tissue release the carbon, nitrogen, and other elements it contained back into the environment. The metals don't decompose. They follow the tissue as it breaks down, return to the soil in forms that may or may not resemble what was originally there, and the contamination re-enters the ground it came from. The harvest didn't happen. The cycle closed back on itself. You start to notice, sitting with this, that phytoremediation is not a biological process that can be separated from the human management around it. The plant accumulates, but accumulation without removal is only relocation, from the soil into the tissue and then back into the soil, perhaps in a different layer, perhaps in a slightly different chemical form, but not gone. Removal requires cutting. Timing the cut matters more than it might seem. Metal concentration in shoot tissue varies across the growing season. In many hyperaccumulators, the highest metal concentrations in leaves occur during active growth, when the plant is pulling water and nutrients upward most rapidly, and the metals are moving with them. Toward the end of the season, as growth slows and some metals may be remobilized downward or redistributed internally, the concentration in harvestable shoot tissue can decrease. Harvesting at peak accumulation, which in many species coincides with peak biomass, maximizes the amount of metal removed per cutting. Some remediation projects harvest twice in a single growing season. If the plant regenerates from the root after the first cut, the second growth period draws additional metals from the soil, and the second harvest removes another portion. The total removal across two cuts can exceed what a single end-of-season harvest would achieve, because the plant is mobilizing metals from the root during the recovery and regrowth period. Still, even well-timed harvests remove only a fraction of the contamination per growing season. In cadmium-contaminated agricultural soils, studies with Thlaspi caerulescens, one of the most studied hyperaccumulators, a small flowering plant in the mustard family, suggest that meaningful reduction in soil cadmium concentration requires multiple growing seasons, often 10 or more depending on the initial contamination level and the depth of the contaminated layer relative to root depth. 10 growing seasons is a decade. Phytoremediation, when it works slowly. This is not a failure of the biology. The biology is doing what biology does, operating as biological timescales, moving material incrementally, season by season, from soil into harvestable tissue. The slowness is real, and it matters for how remediation projects are designed and funded, and for which sites phytoremediation is actually appropriate. A site that needs to be redeveloped within 2 years is not a candidate. A site with moderate contamination, stable ownership, and a decade of patience is a different situation. What happens to the harvested biomass is its own logistical problem. The shoot tissue of a hyperaccumulator after several growing seasons on contaminated soil contains metal concentrations that may qualify it as hazardous waste under various regulatory frameworks. It cannot simply be composted in an ordinary facility, where the metals would be redistributed into the compost product and then applied to other soils. It cannot be left in piles at the site edge. The harvested biomass has to go somewhere, and where it goes determines whether the metals are genuinely removed from the environment or simply moved to a different part of it. Incineration concentrates metals into ash. The ash volume is a small fraction of the original biomass volume. Plant tissue is mostly water and carbon compounds that combust. What remains is a concentrated metal residue that may, depending on the metal content and the regulatory context, qualify for metal recovery, the process of extracting economically valuable metals from the ash for reuse. For nickel hyperaccumulators in particular, the ash from incinerated biomass can contain nickel concentrations comparable to low-grade ore. The plant in this framing becomes a slow-speed mining operation, drawing nickel from soil too diffuse to mine conventionally and concentrating it into a harvestable and then recoverable form. This has a name: phytomining. It exists as a concept and in small-scale demonstration projects. It has not yet become economically significant at scale, partly because the metal market value fluctuates and the economics of biomass collection and processing are not always measurable, partly because the suitable hyperaccumulators have growth rates and biomass yields that make large-scale operations logistically demanding. But the chemistry works. The nickel is there in the ash, recoverable. You start to notice that the plant in this framing is not simply a cleanup tool. It is a concentrating device, doing over seasons what geological processes do over millennia, taking diffuse material from a large volume of soil and bringing it together into a smaller, more manageable form. The cut happens, the biomass leaves the site, the metals go with it. Whether that constitutes remediation or mining or both depends on the site, the metal, the economics, and what the land is needed for next. The biology doesn't make that distinction. It only accumulates. What happens after accumulation is entirely a human question, answered differently in different fields, in different countries, in different seasons. The soil that remains after a harvest is slightly less contaminated than it was, only slightly, and the roots are already reaching into it again. The plant is gone, cut at the stem, biomass removed, metals carried away in the harvested tissue. What remains above the soil is a stub of stem or nothing. What remains below it is a root system. And around that root system, extending outward a few millimeters into the surrounding soil, is the rhizosphere. Still there, still chemically distinct from the bulk soil beyond it, still inhabited. This is where something worth noticing begins. The rhizosphere doesn't end when the shoot does. The organic acids the root released during the growing season have altered the local chemistry in ways that persist. The microbial communities that assembled in response to root exudates, drawn by the carbon compounds the root was releasing, shaped by the pH the root created, influenced by the specific molecules different plant species produce. These communities don't disperse immediately when the plant stops actively growing. They remain in altered form, metabolizing what the root left behind, continuing processes that the root initiated. This matters for what happens to the metals in the soil that the plant didn't take up. Microbes in the rhizosphere interact with metals in several ways. Some bacteria and fungi produce organic acids that increase metal solubility, dissolving metals from mineral surfaces and clay particles, bringing them into soil solution where roots can access them. Some produce siderophores, small molecules originally evolved to capture iron from the environment that can also bind other metal ions. Some reduce metal ions enzymatically, changing their oxidation state and therefore their chemistry, their mobility, their availability to plant roots. Sulfate-reducing bacteria, present in rhizosphere communities of many wetland and flooded soil plants, can produce sulfide ions as a metabolic byproduct. Sulfide reacts with many heavy metals to form insoluble metal sulfides, the same type of chemistry that stabilizes phytochelation metal complexes in the vacuole, now happening in the soil itself. In some contaminated sites, the microbial activity in the rhizosphere effectively immobilizes metals in the soil adjacent to the root, not by removing them, but by converting them to forms that don't move, don't dissolve into soil water, don't travel towards groundwater or into neighbouring soil layers. I tend to think this is underappreciated. The rhizosphere microbiome isn't just a community that benefits the plant during the growing season. It is a biological infrastructure that the plant builds by releasing exudates, by creating chemical gradients, by depositing carbon into the soil through root turnover, and that continues functioning in modified form after the plant itself has been removed. The cleanup, in other words, has two components. The visible one, metal removed in harvested biomass, and the invisible one happening in the soil, mediated by organisms that took up residence because the plant made the conditions for their residence possible. Something in that root turnover contributes to this. During the growing season, plant roots are not static structures. Fine root tips are continuously dying and being replaced, and the carbon they deposit into the soil as they decay feeds microbial activity. In some ecosystems, the annual carbon input from root turnover exceeds the input from decomposing leaves. In the rhizosphere of a hyperaccumulator, this carbon input shapes which microbes thrive and which decline, selecting for communities whose metabolic activities happen to interact with the contamination in ways that are sometimes beneficial to the remediation process and sometimes not. The relationship between rhizosphere microbes and phytoremediation efficiency is bidirectional. Certain rhizosphere bacteria, plant growth promoting rhizobacteria, studied extensively for their roles in agricultural contexts, appear to enhance metal uptake by hyperaccumulators. They produce compounds that increase metal mobility in the soil adjacent to the root, making more metal available for the plant to absorb. They may also reduce plant stress responses to metal load, helping the plant maintain growth rates under contamination conditions. In inoculation experiments, adding specific bacterial strains to the rhizosphere of hyperaccumulators growing in contaminated soil has increased metal accumulation in shoot tissue by measurable amounts. The plant is not remediating alone. It never was. The fungal component of the rhizosphere is less straightforward. Mycorrhizal fungi, symbiotic fungi that colonize plant roots in most terrestrial plant species, extending the root system's effective reach into soil, interact with metal-contaminated soil in complex ways. In some hyperaccumulator species, mycorrhizal colonization is reduced compared to non-accumulating relatives, possibly because the high metal concentrations in the root tissue are harmful to the fungal partner. In others, mycorrhizal fungi appear to buffer metal uptake, reducing the concentration of certain metals that reach the root interior, which is the opposite of what phytoremediation needs. The relationship between mycorrhizal associations and hyperaccumulation is not simple, and the research findings across different species and metals are not consistent. Between seasons, the fungal networks remain in the soil, connected to whatever other plant roots are present, continuing their exchange of carbon and nutrients through the mycelial web. Whether the rhizosphere of the hyperaccumulator, once established, shapes the fungal community in ways that persist across the dormant period and influence the next growing season. This is known to some extent for bacteria and much less clearly for fungi. The dormant root is still releasing compounds into the soil, not at the rate of active growth, but not nothing. The rhizosphere that formed around an active hyperaccumulator retains some of its distinct chemistry even through winter, even through the period between harvest and the next growing season. The microbial community shifts, slows, metabolizes differently in cold or dry conditions, but does not fully reset to the bulk soil community that exists a meter away from the root zone. The soil remembers, not in any way that resembles memory as an experience, but in the way chemistry persists, in the altered pH, the carbon deposits, the microbial community shaped by months of root exudate, the metal speciation changed by that community's metabolic activity. The rhizosphere is written into the soil and doesn't erase cleanly between seasons. What the next growing season finds, when the roots begin extending again from the overwintered root system or from seed, is not the same soil chemistry the first season found. The rhizosphere that forms around the second season's roots forms in soil that has already been biologically altered by the first season's roots. The microbial community is already there, in modified form, ready to be further shaped. The remediation compounds, season by season, quietly in the dark between between harvests, in the chemistry of organisms that have no interest in the project but participate in it anyway. The rhizosphere that formed around a living root is still there, still doing what the living root made possible. There is something quietly interesting about the fact that detecting a problem and surviving it are not the same biological capability, and that in the plant kingdom these two things evolved separately, in different lineages, at different times, and do not reliably appear together in the same species. Most plants can sense heavy metals. This is worth saying clearly before anything else, because it changes how you think about what hyperaccumulators actually are. The capacity to register metal presence, to generate a cellular response when cadmium or zinc or arsenic arrives at concentrations above some threshold, is widespread. It is not the exclusive property of tolerant species. A wheat root encountering cadmium in soil solution will respond at the cellular level to that encounter. Genes will change their expression. Proteins will be produced. The cell is not inert to the signal. What the wheat cell does with that signal, and what happens afterward, is where the divergence begins. In a cadmium-sensitive plant, the cellular response to metal exposure includes production of reactive oxygen species, the same molecules discussed earlier as both signals and potential sources of damage. The reactive oxygen spike activates stress response pathways. Antioxidant systems are upregulated. The cell attempts to manage the oxidative load while simultaneously managing the cadmium load. And in sensitive species, both burdens compound each other in ways that the cell's capacity cannot keep up with for long. The root tip in particular is vulnerable. Root tip cells are actively dividing, and dividing cells are more sensitive to DNA damage than non-dividing cells, because the genome has to be accurately replicated and distributed to daughter cells, and any interference with that replication, including damage from heavy metals binding to DNA or to the proteins that copy it, can result in errors that accumulate across generations of cell division. In sensitive species, cadmium exposure slows root tip cell division, produces abnormal cell shapes in the meristematic zone, and eventually causes root tip death if the exposure is sustained. The root stops growing. The plant's ability to explore soil for water and nutrients is compromised. The cascade that follows is not from the cadmium directly poisoning every cell in the plant. It begins with the loss of the root tip's capacity to extend. Still, in a tolerant species, the same cadmium encounters a different cellular environment at the root tip. The antioxidant capacity is higher. The glutathione pool, the enzyme systems that neutralize reactive oxygen species, the phytochelation synthesis machinery, and the reactive oxygen spike, if it occurs at all, is buffered before it reaches concentrations that would activate the full stress response cascade. The cadmium encounters chelators quickly. The chelated cadmium is moved toward vacuole sequestration. The root tip cell continues dividing. The detection happened in both plants. The cadmium was registered in both. What differed was everything that came after the registration. This is the part that feels, the more you consider it, genuinely surprising. That sensitivity as a biological trait is more conserved than tolerance, distributed more broadly across the plant kingdom, present in species that can do nothing useful with the information the sensitivity provides. The plant knows, in the sense that its cells respond, that something harmful is present, and then it is harmed by it anyway. You wonder, sitting with this, whether sensitivity in the absence of an adequate response is simply a feature of shared ancestry, the detection machinery being old enough that nearly everything inherited it, while the response machinery is newer, more specific, harder to assemble, present only where selection was strong enough and long enough to build it. The genetics suggests something like this. The genes involved in detecting metal stress, transcription factors that respond to metal-induced signals, genes that regulate the expression of stress response proteins, show high similarity across tolerant and sensitive species. The detection system is conserved. What differs is the downstream response. How many transporter proteins are produced, how active the chelation system is, how efficiently the vacuole sequestration runs. These are the components that differ between a plant that manages the detection and a plant that is overwhelmed by what it detected. A related observation, and one that I find genuinely uncertain in its implications. Some sensitive plant species exposed to low sublethal concentrations of heavy metals show a phenomenon called metal-induced tolerance, a partial reversible increase in tolerance following chronic low-level exposure, without the genetic changes that characterize true hyperaccumulators. The plant is responding to prolonged exposure by upregulating some of the same defense mechanisms that are constitutively elevated in tolerant species, as a physiological response rather than a heritable adaptation. The sensitivity is still there, the detection is still happening, but the response is becoming incrementally more adequate. Whether this kind of induced tolerance could, over many generations of plants exposed to low metal concentrations, contribute to heritable changes in tolerance. Whether it represents a pathway, however slow, toward the kind of constitutive tolerance seen in hyperaccumulators is an open question in plant evolutionary biology. The experimental evidence for heritable changes arising from induced tolerance alone is limited and contested. But the question itself reveals something about how the two traits, sensitivity and tolerance, are related at the mechanistic level, using overlapping molecular components, with tolerance appearing to be in part an amplified and constitutively active version of the same response machinery that sensitive plants activate only transiently. The gap between sensing and surviving is a gap in quantity and coordination, not in the fundamental type of biology being performed. A plant in contaminated soil that cannot survive is not failing to detect the problem. It detected the problem correctly. It responded as its biology allowed. The response was not enough, and somewhere in the same soil, a plant that is surviving detected the same problem and responded with more of the same machinery, running faster, running continuously, calibrated over a very long time for exactly this situation. The sensitivity they share feels, from here, almost like a starting point that one lineage built further on and another didn't. Not a difference in kind, but in what came after the first moment of sensing, in the cellular decisions, if you can call them that follow from detection. The contamination arrived. Both plants felt it coming in the way cells feel anything. Only one of them was ready for what came next. Something has to happen to the biomass. This is the part of phytoremediation that exists entirely outside the biology, in the domain of logistics and regulation and economics, and yet it determines whether the biology accomplished anything lasting. The plant grew, the metals moved from soil into tissue, the harvest happened. Now there is a pile, or a truck, or a processing facility containing plant material with metal concentrations that, in some cases, would classify it as hazardous waste under the regulations of the country where the remediation occurred. The pile has to go somewhere, and where it goes is not a simple question. Composting is the first option many people consider, because composting is what you do with plant biomass, and it is the wrong answer here. Composting breaks down organic matter through microbial activity and releases the elements it contains into the compost product. Carbon and nitrogen cycle back into forms useful for plant growth. Metals do the same. They are released from the decomposing tissue and concentrated into the compost, which then gets applied to other soil. The metals move from the contaminated site to wherever the compost is used. The contamination is redistributed, not removed. Landfill is a more contained option. In lined landfills with leachate collection systems, the metals in deposited biomass are physically isolated from the surrounding environment, and the leachate that forms as water moves through the decomposing material is collected and treated before release. This prevents the contamination from reaching groundwater. It doesn't recover the metals or make further use of them. It is containment, similar in principle to the vacuole sequestration the plant performed, but at landscape scale and with engineered rather than biological containment. Still, incineration changes the calculation. Burning the harvested biomass destroys the organic matter and concentrates the metals into ash. The volume reduction is dramatic. Plant tissue is mostly water and carbon compounds, and combustion removes both, leaving a small fraction of the original mass as ash. The metals that were distributed through kilograms of leaf and stem material are now present in a much smaller volume of ash at correspondingly higher concentrations. For certain metals, particularly nickel and zinc, the resulting ash concentrations can be high enough to be economically interesting. Nickel, in particular, has a well-developed global market. It is used in stainless steel, in batteries, in various alloys. Conventional nickel mining extracts ore that may contain 1 to 2% nickel by weight, sometimes less. Ash from incinerated alyssum species, which hyperaccumulate nickel from serpentine soils, has been measured at nickel concentrations of 10 to 15% by weight. The ash is, by the metrics of ore processing, a reasonably rich nickel source. Whether recovering that nickel is economically viable depends on factors that shift with metal prices, energy costs, processing technology, and the scale of the operation. Small remediation projects produce small amounts of biomass, and therefore small amounts of ash with small absolute quantities of metal. The infrastructure required to process it may cost more than the recovered metal is worth. At larger scales, with crops of hyperaccumulators managed specifically for metal recovery rather than site remediation, the economics improve. This is phytomining, and you can feel in that word the shift in framing it represents. The plant is no longer primarily a cleanup agent. It is a harvesting system for diffuse metal deposits, drawing material from soil that is too low in concentration to mine conventionally, and concentrating it into a form that processing can work with. The two framings, remediation and mining, are not incompatible. A contaminated site remediated through hyperaccumulator cultivation over a decade of growing seasons is also, across those seasons, producing biomass that contains recoverable metal. The site is cleaned. The metal is recovered. The recovered metal offsets some of the cost of the operation. In favourable conditions, with the right metal and the right market, the offset could be substantial. Pilot projects exploring this model have been conducted in Europe and elsewhere, primarily with nickel hyperaccumulators in serpentine habitats and with zinc and cadmium hyperaccumulators in industrially contaminated sites. Results are promising at demonstration scale. Commercial scale is not yet established. The gap between demonstration and commercial viability involves questions of biomass logistics, processing infrastructure, and sustained metal market economics that plant biology cannot resolve on its own. What the plant resolved is the chemistry. The nickel is in the ash. That much is certain. The ash itself, after metal recovery, contains residual material, other minerals, silica, remaining trace elements, that has to be managed. Recovery is not complete extraction. What is left after recovery may be cleaner than the original biomass, but is not nothing, and it has its own disposal pathway to follow. You probably already sense where this leads. Every disposal option for hyperaccumulator biomass involves a trade. Landfill trades immediate convenience for long-term containment cost. Incineration trades the energy cost of combustion for volume reduction and metal concentration. Metal recovery from ash trades processing cost for recovered value and reduced final waste volume. Each option moves the metals somewhere. The question is always where and with what degree of finality. No option is perfectly clean. The contamination that entered the plant from the soil exits the plant eventually into some other part of the world's chemistry. The biology captured it. The biology moved it. What happens after that depends on what humans decide to do with what the plant collected. The soil at the remediation site is quieter now than it was. The contamination level is lower measurably after years of harvest. The groundwater risk is reduced. The ecological function of the soil, its capacity to support ordinary plant and animal communities, is slowly returning. That is what was accomplished. The metals are elsewhere now, in an ash pile or a landfill cell or a refined nickel product in a manufacturing supply chain somewhere. Removed from the site, no longer in the soil that was the problem, the plant moved them. Everything after that was human work, and the roots that remain in the ground, still intact below the harvest cut, are already beginning in the slow chemistry of dormancy to reach again. Notice what isn't there yet. The hyperaccumulator has been harvested season after season. The soil cadmium or nickel or arsenic is measurably lower than it was at the start. The remediation, by whatever metrics the project used, has succeeded. The site is ready, or approaching ready, for something else to grow there. And the question of what grows back and in what order, and why that order is what it is, turns out to be a biological question with no simple answer and no predictable timeline. Soil recovery doesn't follow the same sequence as soil contamination. Contamination arrived, in most industrial cases, through a fairly direct process. A smelter deposited fallout over surrounding land. A mine released tailings into adjacent soil. Sewage sludge was applied to agricultural fields over years. The contamination had a source, a direction of movement, a chemistry of deposition. It happened to the soil from outside. Recovery happens from within. The first organisms to change after metal concentrations begin declining are not plants. They are microbes, bacteria and fungi whose population sizes and community compositions were compressed or shifted by the contamination. Some microbial functions are particularly sensitive to heavy metals. Nitrification, the process by which bacteria convert ammonium to nitrate, making nitrogen available in a form most plants can use, is inhibited by cadmium and zinc at concentrations commonly found in contaminated agricultural soil. As metal concentrations fall, nitrification rates recover. The nitrogen cycle, which had been running below its potential, begins to restore itself. The soil becomes more capable of supporting plant growth not because the plants arrived, but because the microbial infrastructure that plant growth depends on rebuilt itself first. Something in this order is worth sitting with quietly. Fungal communities shift more slowly than bacterial ones, in most soils. Mycorrhizal networks, the fungal webs that connect plant roots and facilitate nutrient exchange, were disrupted by contamination, some fungal species eliminated from the local community, others reduced in abundance. Restoration of mycorrhizal communities requires either recolonization from surrounding undisturbed areas, if any exist nearby, or the slow recovery of surviving populations. The rate of this recovery depends on soil chemistry, on whether propagules, spores, hyphal fragments, are present in the soil or arriving from adjacent areas, and on whether the recovering plant community provides suitable host roots for the fungi to colonize. Plants cannot establish efficiently without mycorrhizal partners. Mycorrhizal fungi cannot persist without plant hosts. The recovery of each depends on the recovery of the other, and both depend on the prior recovery of the bacterial communities that influence soil chemistry. The sequence is not a line with a clear beginning. It is several processes running in parallel, each enabling the others, each constrained by the others, the whole system edging toward function through a kind of mutual permission. The first plants that establish in recovering contaminated soil are often not the ones that will dominate the site eventually. They're early colonizers, species with small seeds that disperse widely, that can germinate in disturbed, nutrient-poor conditions, that grow fast enough to establish before competition closes in. Ruderol species in botanical terminology. Plants adapted to the edge of things, to places that are not yet stable. In heavily contaminated sites where remediation has been partial, these early colonizers may themselves show some metal tolerance, not hyperaccumulation, but enough resistance to survive in soil where the contamination has been reduced but not eliminated. Their presence further alters the soil. Their roots contribute organic matter. Their rhizospheres support microbial activity. Their litter, when they die, adds carbon to a soil that is often carbon depleted after contamination and the years of remediation cropping. I tend to think this phase is underappreciated in how remediation success is measured. Success is usually measured in metal concentration. The soil is below a regulatory threshold. The remediation is complete. But the soil that meets the metal threshold may still be biologically impoverished in ways the threshold doesn't capture. The microbial community may be recovering but not recovered. The mycorrhizal network may be sparse, the organic matter content may be low, the soil structure, the aggregates of mineral particles and organic matter that give healthy soil its texture and water-holding capacity, may have been degraded by years of industrial contamination and then by the repeated mechanical disturbance of hyperaccumulator cultivation and harvest. A soil that passes a metal test is not the same as a functional soil. What eventually grows on recovered ground reflects the biological state of the soil at the time of establishment, which reflects the history of contamination, the depth and duration of remediation, the management of the site afterward, and the availability of colonizing species from surrounding areas. In some cases, the plant community that establishes looks nothing like what was there before contamination. Different species, different proportion, a community assembled from whatever arrived and could establish in the conditions that existed at recovery, rather than a restoration of the original. In other cases, where contamination was less severe or remediation more thorough, recovery curves back towards something resembling the pre-contamination community. If records or reference sites exist to indicate what that was, neither outcome is failure. Biology doesn't fail at recovery. It does what the conditions allow, assembles what can assemble, and the resulting community is functional in the ways a community in those conditions can be functional. Whether it matches a historical baseline is a question humans bring to the soil. The soil itself has no attachment to what it supported before. The ground that was written off, too contaminated, too degraded, too far from what soil is supposed to be, begins again. Not from nothing, from what remained, from the microbial populations that survived at reduced levels through the contamination, from the seeds that arrived on wind from undisturbed areas nearby, from the mycorrhizal spores in the soil, dormant, waiting for roots to reach them. The first seedling that establishes in recovered ground doesn't know what the ground went through. It finds chill. It sends a root down. The root encounters the rhizosphere chemistry left by years of hyperaccumulator cultivation, altered, inhabited, biologically active in ways the raw contaminated soil was not. The seedling establishes more easily than it would have in the original contaminated soil. More easily, perhaps, than it would have in soil that had simply been left alone. The remediation made the ground ready for something it couldn't hold before. And the soil that was shaped by all of this, by the contamination and the hyperaccumulators and the harvests and the microbial recovery and the early colonizers, is not the soil that was there before any of it happened. It is something that came after. A soil with a history, carrying the chemical memory of what it went through, now supporting whatever can grow in the conditions that history created. The first root of the new community reaches down into it, and the ground, which has been doing this, receiving roots, releasing what it holds, being slowly changed by what grows in it, the ground simply continues. And that's where we'll rest tonight, in the quiet recovery of ground that was given back to itself, slowly, season by season, root by root. If this journey through contaminated soil and patient plants moved something in you, alike means more than you might think. It helps this channel reach other people who listen the same way you do. If you're a member or have supported through Super Thanks, thank you genuinely. This work exists because of that. And if you have a thought, a question, or simply want to share where in the world you listen from tonight, the comments are a calm place. Always read, sleep well. The ground is still working, so are you.